Alrighty, so antimicrobial medications. We're going to start by looking at some of the history and development. And so the first successful documented use of salvaricin was used by Paul Ehrlich on syphilis. And it was the first documented use of antimicrobial medications. So that's the first time that we saw that use of microbial control within the body on an infection. Prontosil was the next documented chemical prescribed to be effective. It was used against streptococcal infections in animals. Alexander Fleming he actually did produce it in a way, discovered that the mold Penicillium produced a bacterial-killing substance that he called penicillin. And basically what happened was he had plates of bacteria that he was trying to grow. They had been left exposed for too long to the air, and the mold landed on the dish, and then he kind of let the dishes sit for a while, kind of like I've been known to do. And those dishes, all of a sudden, he noticed that the bacteria disappeared, where the fungus proliferated. It grew. And then all of a sudden, the areas surrounding the penicillium, the bacteria was dying off. So he could see what we now know is the zone of inhibition around the mold. And that's basically because the substance that was being produced is actually an enzyme. If you remember much from biology, fungus secrete enzymes into the environment. The enzymes digest organic material, and then the fungus absorbs. There is no ingestion. So penicillin was the very first antibiotic that was naturally produced by a microorganism. So this is very similar to what he saw. You could see that there are streaks of bacteria on the agar plate and then a large colony of mold that is growing. And you can see where the streaks of bacteria have disappeared. You can see that zone of inhibition very, very clearly around this area here. There. So it's easy to identify an antibiotic-producing microorganism. The hard part is finding the antibiotics that are sufficiently known as selectively toxic. Anywho. So post-microbe, modified antibiotics are actually referred to as semi-synthetic antibiotics. Selective toxicity means that the agent is more toxic against bacteria than against humans. You want selective toxicity. You don't want to take an antibiotic that's going to attack you and destroy your cells as well as the fungal cells, right? Or I'm sorry, not the fungal, but the bacteria that you're infected by. That's kind of important, right? You can say yes. Yeah. Nice. Okay. So Selma Waxman later showed that Streptomyces griseus, a soil bacteria, produced an antibiotic called streptomycin. How many people have ever taken streptomycin? A couple of hands went up. So the chemical structures of drugs like penicillin G can be altered to give them new properties. So that is one of the reasons why we have been able to develop new antibiotics. For example, penicillin G, which is the most active against gram-positive bacteria, can be altered to produce ampicillin, which can kill additional gram-negative species as well. So another antibiotic, methicillin, can be used to treat infections caused by penicillin-resistant organisms. Today, a variety of different penicillin-like medications exist and form what we now know as the family of penicillins. Now, as you're writing things in your note packet, I want you to think about who often gets prescribed penicillin. How much experience have you had in taking antibiotics? A good amount? A decent amount? So if you have taken a lot of antibiotics, when was the last time you actually took penicillin? Ear infection, strep throat. I'm kind of starting to give it away a little bit. So maybe, so you said 7th grade, right? But like even for strep throat, if you've recently had strep throat, did you take penicillin? You did? If you did, I'm kind of surprised. Have you taken ampicillin or amoxicillin? Okay, so here's the thing. Most individuals that take penicillin are usually under the age of 2. After that, penicillin doesn't work very much anymore. Why do you suppose that happens? They become resistant to it. So that population ends up with the gene or that R plasmid that codes for the production of resistance to penicillin. But we have now found the way to change penicillin in such a way that we can create another variation that resistance gene does not work against. But eventually then the population will become resistant to that as well. So let's take a look at some of the antimicrobial chemotherapy terms. So you hear the word chemotherapy and what do you think of? You think of cancer. Chemotherapy is really just using chemicals as a therapy. Sometimes you take just one, sometimes you take a combination. It depends on what's wrong with you. But when you look at these terms, look at antibacterial drug versus antibiotic. An antimicrobial drug is used to treat a disease caused by bacteria, whereas an antibiotic is a compound naturally produced by certain molds and bacteria that inhibits the growth of or kills other microorganisms. So what's the difference? Antibacterial drug versus antibiotic.
Antibiotic is natural and it's based off natural things, and antimicrobial is chemicals.
Yep, you're right. So an antibiotic is produced by another living organism. So it's natural, whereas an antibacterial drug might not be. It might be synthetic. So an antiviral drug, how is that different than an antibacterial drug?
It's the actual virus that you have, but it's like changed, right?
Well, it's not like you're ingesting or taking a drug that contains a virus, but it's a drug that attacks a virus. So it's the same thing as an antibacterial. Antibacterial attacks bacteria only. Antiviral attacks viruses only. So what's a bacteriostatic drug? Is it going to kill the bacteria? Is it going to get rid of the infection for you? Just stops it, prevents it from growing. Bacteria are still there. It just prevents them from increasing their numbers in their population.
So after you take that, would you take like an antibiotic to get rid of them?
It all depends on the infection. We're going to talk more about like what you would take under what circumstances later on here in the notes.
Could bacteriostatic, could the bacteria become resistant to that too?
I would expect so, yes. So a broad spectrum antimicrobial. Think about it because you've probably all used one of these. Is an antimicrobial that's effective against a wide range of microorganisms, often including both gram-positive and gram-negative bacteria. A broad-spectrum antimicrobial. You fall down, you get a boo-boo. You clean it off. What do you put on it before you put the Band-Aid on? Neosporin. Neosporin is a broad-spectrum antimicrobial. Yep. Okay. All right. So the rest of these terms are what we're going to be looking at as we move a little bit farther into our notes. So, in terms of antimicrobial drugs, most modern antibiotics come from microorganisms that normally reside in the soil, believe it or not. Who'd have thought? Everybody says, don't eat dirt. Well, guess what? Dirt has a lot of good stuff in there. A little bit of dirt isn't going to hurt. Okay? I've been around my cousin when she had her child and, you know, other family members and friends who have had babies, and, oh my gosh, the Nook dropped on the floor. Well, really? It's not the end of the world. Okay? Probably going to be healthier if you ingest a little bit of dirt over your lifetime. So bacteria like Streptomyces and Bacillus are going to be two groups. And then in terms of fungus, Penicillium and Cephalosporium are two fungi groups that produce antibiotics. Current antimicrobials at this point are created through a combination of bioculture and chemical modification, so that makes them semi-synthetic. Okay? The chemically modified compounds are referred to as semi-synthetic, and in some cases, the entire drug is entirely synthesized right in a lab. Kind of interesting. They'll take the bacteria and they'll be just everything will be grown directly in a lab, but it's still considered semi-synthetic because of the use of the microbes. So these partially or totally synthetic chemicals are still called antibiotics because microorganisms still naturally produce them, even though humans might be controlling their reproduction and, you know, culturing them on purpose. All right. So this is what the family tree of penicillins looks like. And you can see down at the bottom of the screen that penicillin G and penicillin V were the first two that were developed. And that was in the 1940s. After that, you can see that the tree branches a lot. And that's because in the late 1950s, we learned how to make those adjustments. So you can see ampicillin, which is one that you might be familiar with. You see methicillin, you may have heard of that one. But as you look at the rest of the tree, are there others that you recognize? Has anybody been on any of those that are listed? So antibiotics are chemotherapeutic antimicrobials that are isolated from microorganisms. Okay? All right. So this is just a table showing some of the different species of bacteria and fungi and which of the antibiotics come from each of those species. So I don't know what you've been exposed to, like what you've taken. I know that bacitracin is something that is very common, sometimes in households. Bacitracin can be found in like an ointment that you use, kind of like Neosporin, that then you can apply to wounds, and it's kind of a general antimicrobial. But most of what we have probably recognized in microbiology has been streptomycin, Kanamycin was one of the antibiotics that we looked at in terms of resistance earlier in first semester. We watched those video clips of that lab that we couldn't do because it involved human pathogens. Do you remember that? About the kids were demonstrating how they did the lab. So, what are the features of antimicrobial drugs? There's a lot of features that we're going to look at. So these are all the things that you would have to take into consideration. If you are thinking about going into pharmacy, if you're thinking about going into the medical field and you are responsible for writing prescriptions, these are all of the things that you would have to take into consideration. Remember when we looked at microbial control, we were looking at those different chemicals, whether it was a sterilant or a disinfectant, etc. And we had to take in certain things, take into certain considerations regarding which type of chemical we were going to use. How toxic was it? How many microbes were present? What kind of microbes are present? How big was the area that we have to clean up or disinfect or sterilize? What was the purpose? So now when we look at antimicrobial drugs, we have to look at some of those same considerations. So I want you to kind of relate what we've already studied to what this current unit is about. So in terms of selective toxicity, antimicrobial drugs exhibit selective toxicity, causing greater harm to microorganisms than to the human host. That's a good thing for us. They do so by interfering with essential biological structures or processes, and we're going to talk about those later on in our notes, that are common in microorganisms but not in human cells. So selective toxicity is a relative term. Antimicrobials that have a high therapeutic index are less toxic to patients. High therapeutic index are less toxic to patients. So the example is penicillin G, which interferes with bacterial cell wall synthesis. It has a very high therapeutic index. So the question is why doesn't this harm a human host? How come it's not dangerous for us to take penicillin? We have different biological structures. If penicillin is attacking or preventing a new cell wall from forming, what's different about our cells from bacteria? We don't have cell walls. So that's why it's a high therapeutic index. So this is kind of my way of trying to indicate like the difference. A small therapeutic index, a small ratio, is very dangerous to the host. It kind of has a blurred line between being toxic and nearly like having a lot of major side effects in the host, as well as being therapeutic and treating the infection. Moderate, it's maybe not quite as toxic to the human host, but maybe it doesn't have as much therapeutic effect either. Where the high ratio, like the penicillin G, is not toxic nearly at all. Some people have experienced allergic reactions to it, but that's about the severity of the side effects. But also has a pretty good therapy rate. Yeah. So obviously, that high ratio is the most desirable. A high therapeutic index is most desirable. The second consideration is the action of the antimicrobial. So we've looked at these terms before in that microbial control unit. an antimicrobial drug may either kill organisms and be bactericidal, or they inhibit their growth and they are bacteriostatic. Now, one way to remember the difference between those two terms is the suffixes. So cidal, homicidal would mean, right, murder, death, killing. Static. If the population remains static, that means it's not increasing, but it's also not decreasing. They're hitting a plateau in their population numbers because of taking that antimicrobial drug. Now, bacteriostatic drugs depend on the normal host defenses to kill or eliminate the pathogen after its growth has been inhibited. So one example are sulfa drugs. Sulfa-type drugs are frequently, prescribed for urinary tract infections, but then your immune system has to get rid of the bacteria on its own. Bactericidal drugs are particularly useful in situations where normal host defenses cannot be relied on to remove or destroy the pathogens. And a given drug can be bactericidal in one situation, and then in another, it might be bacteriostatic. So in looking at a drug to prescribe, you have to pay attention to the different environments or what conditions you're taking that drug, and then that would determine what effect it's going to actually have on the infection that you're trying to treat. Plus the growth stage of the microbe. So where are they at in their invasion of the human body? So the spectrum of activity is the third consideration. Antimicrobial drugs vary with respect to the range of microorganisms they kill or inhibit. So some kill only gram-positive bacteria. Some kill both gram-positive and gram-negative organisms. That's referred to as being broad spectrum, as opposed to killing only a certain type, and that's narrow spectrum. The broad spectrum antimicrobials are very important in the treatment of acute life-threatening infections, as there's no time to culture and identify the disease-causing agent. So a lot of times you go in and you're exhibiting illness symptoms, and the doctor says, well, we're going to take a swab and we're going to culture it. And so they do that, but how many people have had like the rapid strep test done, where they do it like right then and there, and they either get a positive or a negative identification. But you could get a negative identification, and they might say, we don't think it's strep, but they put you on an antibiotic anyways. And then you go home because they want you to start on it right away, just in case, and then they wait for that 24-hour test to come back. And that 24-hour test was the swab that they cultured, grew on a plate, and then somebody in the lab took the plate out of the incubator and looked at it, tested it with hydrogen peroxide. If it bubbles, it's strep. Okay? So that's really all they do. And then they're able to tell you, yeah, it's strep. Okay? So then if you're already on the antibiotics, that's good, because then it hasn't had another 24 hours to grow and incubate and get worse. The disadvantage of a broad spectrum antimicrobial is that they disrupt the normal flora, which in turn then kind of predisposes you to other infections. So the use of narrow spectrum antimicrobials requires identification of the pathogen. That right there is super duper important, okay? That right there. But they cause less disruption to the normal flora. So that's of major benefit. So when we look at the spectrum of activity, check this out. We've got the mycobacteria. They're in a different group because they don't have a cell wall. So penicillin, notice how penicillin doesn't work on them. There's gram-negative bacteria, gram-positive bacteria, the chlamydias and the rickettses. Penicillins work mostly on gram-positive, a little bit of gram-negative, and some of the chlamydias. Which of these antimicrobials on here would you say is a broad spectrum? Tetracycline. Tetracycline. Which one would you consider a narrow spectrum? Isoniazid or a polymyxin. Yep. those two would be definite narrow spectrums. Good.
What were mycobacteria again? Is that just like-
Oh, mycobacteria are the bacteria that don't have a cell wall. So when if the antibiotic that you're using targets cell wall production
so that's what we
or
have.
destroys the cell wall, it doesn't make sense to give somebody that's got a mycobacterium infection
Oh, that.
like a penicillin.
So that's basically what we are.
No, no, because we're not bacteria.
I know, yeah, true. I think that's cells.
Yeah.
Just cells.
Yeah.
Yeah, they're cells.
So, tetracycline is a wide or broad spectrum antimicrobial, and a polymyxin is an example of a narrow spectrum. Good. So, the thing that we have to remember is that a broad spectrum antibiotic are useful if one must begin treatment before identifying either a pathogen or its antibiotic susceptibility. So we have to be careful with that because they can be a little bit toxic. However, broad spectrum antibiotics are more likely to destroy the normal flora, resulting in what we know as a super infection. That's what it's termed. So you still you might have an infection caused by bacteria or whatever the case may be, but then because of the side effects or the destruction of your normal flora, there is no natural competition within your body, and then all of a sudden you start to come down with other infections, or yeah, we'll just leave it at that. Other infections that then you've got multiple things going on in your body, and that can cause kind of overload on the body. All right. Fourth is tissue distribution, metabolism, and the excretion of a drug. Doesn't sound like a very interesting topic, but it kind of is, because I think you've probably all maybe had a little bit of experience, maybe not to the extent of some of the examples that I'm giving you here. So only some drugs are actually able to cross from the blood into the cerebrospinal fluid. So if somebody has an infection in the cerebrospinal fluid, like meningitis, attacks that fluid that surrounds the brain, you have to be very careful about what drug the person takes. Drugs that are unstable in acid are destroyed in the stomach when taken orally. So you couldn't give them something in a pill form. They'd have to get it as an injection, right? You don't want it to go through the stomach if acid's going to impact it. So the half-life of a drug is probably the most important thing to take into consideration, and that's the time it takes for the body to eliminate 1/2 of the original dosage in the serum. And that's going to dictate the frequency of the doses required in order to maintain an effective level. Because if there's not enough there, and there's too many of the bacteria or viruses present, it's not going to have an impact. And then you're taking drugs for the purpose of, well, not for any purpose, really. So one example is penicillin V has a shorter half-life and needs to be taken 4 times a day, whereas azithromycin, with a half-life of over 24 hours, is taken only once in a day or even less. Kind of nice. People with kidney or liver dysfunction often excrete or metabolize drugs more slowly, and so their drug doses have to be adjusted accordingly. So you have to take that into consideration. So this is one of the reasons why I thought it would be interesting for us to take a look at this, and that's mainly because if you're thinking about maybe going into pharmacy, it's a great career. It's a great occupation. You can make a lot of money, but you have to have a good chemistry background. You have to understand all the drug interactions, as well as all of this information that we're talking about in these notes. So this is for ampicillin. And so this is what the concentration looks like in the body over time. So the graph is showing us how many micrograms there are per liter of the ampicillin in the blood plasma. And so notice how here's the MIC, the minimum inhibitory concentration. Do you remember that from microbial control? Maybe not. We maybe skipped that part. That might have been that part that we skipped. The minimum inhibitory concentration for E. coli and for strep is the top line, the larger dashed line. The bottom line is for strep. So if somebody was taking ampicillin, right, they take the dosage initially, And initially, the dosage, right, you take it in, and you're going to have a high level, looks like around 14 micrograms per liter. But then over time, the body starts to break it down, right? And then it decreases. I'm having a hard time doing this. And at a certain point, it drops below the minimum inhibitory concentration for E. coli. So if somebody had an E. coli infection, ampicillin taken every 8 hours isn't going to be effective. They have to take it probably a little bit every 4 hours. But if somebody had strep, could they take ampicillin every 8 hours? So you can see that if the dosage is taken here at 8 hours, the individual goes back up, and that concentration in the blood plasma doesn't ever drop below the MIC for strep, whereas it does for E. coli. So that means you'd have to prescribe it at 3 doses a day, so every 8 hours. And if you wanted them to take it for 10 days, you'd have to give them 30 tablets. Okay? This is the last one that I wanted to talk about in this lecture, and that's going to be the effect of combinations of antimicrobial drugs. So sometimes combinations of antimicrobial drugs are used to treat infections, but care must be taken as some drugs will counteract the effect of others. So we can look at the interactions of drugs and identify that they would fall under one of 3 categories. The first one being synergistic. So when the action of one drug actually enhances the activity of another, that combination is called synergistic. Makes sense. It sounds like synergism, that relationship that exists between organisms in ecology. In contrast, the opposite is when the activity of one drug interferes with the activity of another, and that's called antagonistic. Makes sense, right? But if the combination is neither synergistic nor antagonistic, they're called additive. So it might be that each drug acts on its own in a different way, but it doesn't improve the action of the other drug that's in its presence, but neither does it interfere with it. Maybe the individual is taking 2 drugs that are attacking 2 different mechanisms of, for example, bacterial reproduction or just metabolism.